Hello and welcome everyone in another episode of Researcher's Celebrity. Today we have Dr. Narayan Komaravelli with us. A brief about him is he has done his bachelor's from Osmania University in life sciences, master's from BITS Pilani and in collaboration with Shankar Netralaya in medical laboratory technology. PhD was also again a collaborated program with Bharat Biotech and Jawaharlal Nehru Technological University from Hyderabad. Then he moved for his postdoc at National Institute of Immunology, Delhi. Did some research scientist work at Orchid Chemicals and Pharmaceutical at Chennai, and then moved United States of America for pursuing his postdoc as well as research scientist. He was there at UTMB Galveston, and he was working with infectious disease and neurobiology. Presently, Dr. Narayan is clinical trial manager at a global CRO. With this brief introduction, I would like to welcome Dr. Narayan at the platform.
Hello, everyone. I'm glad to be here, and thanks, Raja, for this wonderful opportunity to meet you all.
Yes, thank you and welcome. And now the conversation, all our researcher celebrity conversation starts with how and when you decided that you wanted to be a researcher.
That is a tricky question. I could say my innate ability, or my childhood from many ways, I was always interested in science. So that curiosity, my nature itself is curiosity. I'm always curious and wonder about things. Around the world, especially science and technology, which has any touch. Not only in biology, in general, I'm passionate about science and technology. How all this science is helpful for improving not only the health, but improving the life and quality of the people. Say, for example, even our TV has an entertainment. It improved people's life, and from medicals, it improved people's quality of life. So my family had a traditional business of a medical shop. We always associated with the doctors. And I was curious, how patients, doctors see the patients and they treat medicine and they will come. I used to help my dad and uncles. I used to sit in medical shop. So I used to wonder, well, how do people go to doctor and doctor writes some medicine and we used to sell. So I wanted to originally become doctor. I could not. That is a different story. But I always wonder how this medicine are manufactured and what kind of medicine are there. So that led me to curious. So when I was in BSc, I realized there are not only chemical medicals, there are also biological medicals. Biologics are there. So that led me to further pursue my career in molecular biology side. And so that's how I ended up in doing my, after BS, I did my master's. I wrote this JNU central entrance, but somehow I could not get it. So whatever I got a couple of, not couple, actually, I have about 4 or 5 opportunities. One at Madurai Kamaraj University. I got MSc Biotechnology position there, and this Shankar Netralaya program, which is called MS with collaboration with BITS Pilani, Medical Laboratory Technology program. So I was debating between these two courses, including MS MS Genetics and Microbiology at Osmania University. So the decision made was kind of funny. I didn't want to do in Hyderabad because I wanted to have more exposure to myself. So I wanted to go away from Hyderabad. So their options left with some of them in Gwalior and some of them in Delhi. So I didn't want to go too far also from Hyderabad. So I eliminated my Gwalior option and Delhi option. So left with Chennai and Madurai Kamaraj. And Madurai Kamaraj is a small city, very small city. So I wanted to ended up in a big city like Chennai. and also it's close to Hyderabad. And also, I had a discussion with my microbiology professor at my BSc college, and she suggested, "Hey, look, don't go with the degree names. I mean, medical laboratory technology. It sounds not great, but the curriculum is great. I mean, it's a 3 years course and a 2 years college course, and 1 year is a research internship project." So, and Shankar Nethralaya is that, I mean, even now it is great, but that time it is one of their technologies and their labs are one of the best in country. I mean, even in that college, Shankar Nethralaya itself is largest eye hospital in Asia, even till now, from that time to now. So they have world-reputed doctors, and their curriculum is great. I mean, they taught all the hematology, pathology, physiology, anatomy, chemistry, biochemistry, microbiology, genetics, all related to medical lab and I mean, medical diseases and the diagnostics. It is so she said Madurai Kamala University MS Biotechnology, they just focus on biotechnology, and this is though Bits Pilani and Shankar Nethralaya both are great names, so you should consider that. And also, I had a friend, my BSc friend, who also took that course. So I thought it's a company also, but I'm very grateful that I did that course. I'm grateful for her professors. Always, you go. I mean, went with advices of your peers and professors who, I mean, they always have better knowledge than you. So I took her advice and I went there and I completed the course. I'm very glad I did that. I mean, it's a 3 years course, and the final year I did 1 year internship project in molecular analysis of retinoblastoma. That time it was a DNA sequencing by using a 8 capillary DNA ABA machine. To be fact, that is one of the only few machines in India. That time, handful, we can count fingers. I mean, if not even 10 machines were there in whole India. A couple of them in CCMB, couple of them in one in NII, one in ICGEB, and two in IISc. Total about not even 10 machines are there. That time I did this course, and even from my dissertation, there were 2 publications also. So I'm very happy I did that course.
So when you were experiencing the research in hospitals and at the university,
yes,
what was the clicking factor that you wanted to do a PhD but not directly join the research in industry?
Yeah, that is again, it is a no-brainer question, because I wanted to proceed my career further in somewhere where I have a major impact in scientific publication wide, and also I wanted some of my research should help people. I mean, some product it should come or something like that. I mean, so that's why I want. So I realized just with, I mean, my course is called MS. MS is not enough. I mean, I had another idea also that time to open a diagnostic center where also I would help people and also help doctors to get diagnosis and treatments and cure. But I was with my, again, discussion with my friends and professors, we realized MS is not enough. I can do if any diagnostic center after PhD would be great. So at the time of to have more impact on science and all that, PhD is necessary. That is what our curriculum says. I mean, without PhD, you cannot do your own research, right? I mean, and apart from that, that glamorous name to have doctor in front of you, that is also, I mean, there are so many factors. I mean, without PhD is a necessary step to have anything in further in our career, right? So that's why I did PhD.
Nice. So let's talk about the journey of PhD.
Oh, that itself, it's again There are so many tricky, funny things happen. So I realized to do a PhD. I thought of going abroad also. I mean, I did it in. I got a position in UK and Australia also, but they had some financial situation. They asked money for that to pay. I mean, it's an MS and PhD program. So I already did master, so I didn't want to do. Master. So they said I need to pay more money for that for PhD. Those are nice universities, but somehow I didn't get understand why for PhD we need to pay money. But they said once I pay money, they also give me internship. So whatever those things I did not understand. So I had prepared for my CSR, JRF, and all that. I qualified for CSR, JRF. Um, but before doing that, I wanted to do specifically the research in molecular biology of DNA protein interactions in gene expression in oncology.
Mm-hmm.
So when I said that, everywhere, wherever I went, people were saying you cannot be too specific on those projects.
Mm-hmm.
You have to take whatever you get.
Mm-hmm.
So I was looking for those. I mean, because of my limited knowledge that time, I could not find exactly professors who were doing that. So then I realized that time they used to call CBT, Center for Biochemical Technology, and now it is called IGIB, Institute of Genomics and Integrative Biology at Delhi. So I wanted to do my PhD either NII or IISc. These two places only I wanted to do, or ICGEB Delhi. But unfortunately, none of these places I didn't get. I wrote entrances, but I did not get. Then somebody at NII suggested me IGIB is also very upcoming place. Doctor that time director was Dr. Samir Brahmachari, and he was a CS. I mean, later he promoted to CSR DG, and he retired. So you should go to him, and he's also like. I mean, I guess your mentality looks similar with him. He also always dream a lot.
Mm-hmm.
You should go to him. So I really connected to that guy. I mean, I don't remember even that person now, an AI person. So I went there, but because I did not get any appointment to him, so I looked into his group. So one faculty, I wrote an email that, hey, I mean, that faculty is from Chennai area. So I wrote, hey, I came all the way from Shankar Nethralaya, Chennai to meet him. So can you help me? I wanted to do a PhD under Dr. Brahmachari. So he liked that idea all the way. I came from Chennai. So I said, I'm staying 3 days. I mean, I came to write an entrance, but I'm staying here 5 days. I did not get my second round of interview. So, anyways, I'm here for 3 more days. So, can you give an appointment so I can discuss with you?
Mm-hmm.
So he said he asked me to come, but unfortunately on that day he was down with fever. So he recommended some other guy in Brahmachari group.
Mm-hmm.
I talked to his name is Dr. Devashish. I forgot last name. So you meet him. I'll recommend to him to at least help him to do further guide. So I met this guy, and then he put a word to Brahmachari. So Dr. Brahmachari said, "We'll see. Let him ask him to wait. I'm super busy. We'll see if he wait till evening. We will. I'll think of about him." So
Mm-hmm.
you can talk to him, and you and there is so Brahmachari. Dr. Brahmachari lab itself is named called Functional Genomics. So he has two labs. One is a dry lab, one is called wet lab. So wet lab they do all molecular biology and genomics experiments, and in dry lab they do bioinformatics and sequence analysis and all that. So Dr. Devashish interviewed me. I mean, he had a discussion. He didn't say it's an interview and all that. So he gave me so many tricky questions in maths, but I didn't know the formulas. But I somehow answered it. So he asked me, "How did you answer?" I said, I'm not major in maths. I studied maths till 10th class only. But somehow by eliminating and using, I answered his questions about. I got about 80% of his score. I mean, he expected only 50%, but I got 80%. So he was very happy. Then he introduced to another professor, another faculty in Dr. Brahmachari's group. Her name is Dr. Mithali Mukherjee, I still remember. So she did again interview in, I mean, she wondered how come I did medical laboratory technology and did 3 years of courses. I mean, she asked questions in biochemistry, genetics, molecular biology, microbiology, whatever she knows. So I answered all of her questions and I gave more thought-provoking questions also to her. And she also very impressed, and they both of them gave very positive note to Dr. Brahmachari. And Dr. Brahmachari met me at evening 8 o'clock. I mean, I was there. I went at morning 9 o'clock, and I went, but he was not there. He came at 10 o'clock or 11 o'clock, and he went in once in a while. He went out also for other meetings, and somebody said he's still waiting for you. So he came, and he was very impressed with that. I really liked your patience and your endurance. You came without appointment and you waited for this long. So he didn't ask me any questions. He didn't interview also. He said, my both of my left hand and right hand liked you. So there is nothing need to ask. So by that time, my CSR JRF results did not come. So he said, don't worry about your CSR results. I can take you under my quota. So if you get fellowship, fine. Otherwise, also I can pay you. So we started program. I was there about 6, 8 months. Then somehow I didn't like that atmosphere there. Dr. Brahmachari is great, but he's so busy. He would never meet as a PhD student. Then I asked. He said, "Oh, I don't have time to meet you and guide you. You have to listen to Dr. Mithali and Dr. Devashish, whatever they say, do it." And maybe in second or third year, when you have a one publication or two publications and something is going your project, that time I will interview. So that idea I did not like. I thought, oh, maybe in this way I will complete PhD only after 7, 8 years. And so I didn't like the daily atmosphere for some reason. I mean, I used to get sick all the time. So, I mean, these are the practical reasons. There
Yes.
is no, it is nothing that I didn't like scientifically there, but I didn't get the topic, whatever I interested in, like regulation of gene expression part. Or, I mean, they asked me to always do some or the other thing, and they say, don't worry about the project. You do 3, 4 projects, and whatever is going through, we will publish it. I mean, you can submit as a thesis and you can get PhD. And then in that way, how many years it will take? Then they didn't like that question. Dr. Mitali was so serious. No, in this lab, you cannot ask me how many years it will take. You keep doing that. We take only people who have passion to the science. So we see a passion in you. That's why we took. But if you worry about the length of the years, oh, this is not the lab. You had to stay here. So I didn't understand. I mean, I thought maybe she's indirectly asking me to leave or something. I mean, at that time when we come from freshly from masters into PhD, so young and hot-blooded people. So I took it as a very offensive. I thought maybe she's asking me to leave. So next day I went and I put resignation to her, to Dr. Brahmachari, and they said, "No, I didn't ask you to leave," she said. I mean, you said that this is not the lab for you. What does that mean? So she said, "No, I didn't mean it that way. I mean, you need to look for other projects, or you have to like whatever we are doing." But then she said, "What would you do?" "I don't know. I'll go back to my home and I'll see whatever I'll do." So then I went to Hyderabad, and that time I have a couple of friends, nice friends I made in IGIB. So one guy wrote an email, hey, there is one guy called Dr. Khatri. He is on deputation from IGIB to Bharat Biotech. So Bharat Biotech was a very new company, startup company that time. So they hired Dr. Khatri from IGIB as on a deputation from CSIR. IGIB transferred few technologies to Bharat Biotech like streptokinase, lysostatin, and EGF, and HGH, and this kind of technology they transferred. And they helped also. He helped also them to clone. Their first commercial product is hepatitis B vaccine. So he helped them clone. They bought commercial technology from some whoever had patent rights. So technology, once the rights are over, they started their vaccine as a first product. So
Nice.
we helped them to as a scientifically how to proceed and all that. So they liked him, and they put a proposition to him that why don't you stay Bharat Biotech and you develop whatever some commercial products. So then he agreed for that, but he told Bharat Biotech that I would like to work. In my life, I always worked with PhD students or postdocs. So I don't want to work in industry just like that. So I want to work with PhD students. So that they will work with passion. Also our products will be also made. It is a win-win situation for everyone. So in fact, Bharat Biotech's owner, Dr. Krishna, he liked it very much because he himself is a scientist. So he said, okay, go ahead, do whatever you like it. So then a friend of mine at IGIB suggested, hey, Dr. Khatri is a very famous personality. He accomplished a lot. He had a lot of patents, and he was one of the person in India who got first cell publication. In 1960s. Maybe 19, not 60s, 70s or 80s. From his postdoc, he got first two cell publications and bunch of PNAS publications. So I looked his profile and I liked him a lot. So I went and approached him. I wanted to. My friend suggested that you're here, and also I'm looking for completing my PhD. So they said he did interview and he liked me. He took immediately to Dr. Krishnal, and they all interviewed me, and they liked me, and they said, okay, we can give PhD and you can start a program. So meanwhile, I also did a course in bioinformatics. During that my free period at GV Bio. So I learned how to analyze these sequences and identify drug candidates and all those kind of stuff. So Dr. Khatri liked it, and so my initial project started with. It's a vaccine. So by that time, Bharat Biotech had lysostatin. I think it has not yet commercialized. It never commercialized. Maybe some intrinsic issues. So this is a tropical. Their goal is to this lysostatin is a molecule that kills Staphylococcus aureus. But the thing is, it works only on tropical. Not be injected by people. So we got idea from that. He asked me what Dr. Khatri and I we decided, hey, why don't we look at novel vaccine candidates? So he suggested to look at Staphylococcus because there is no vaccine for Staphylococcus. Even there is no vaccine. So I really liked it. It's a challenging project, though I wanted to be in oncology. So they said, no, we don't do oncology. But, by that time I kind of beaten. I kind of, I tried stuff, and I wanted to go always into oncology, but I did not get what I want. So I kind of got disappointed and I disheartened. So he at least helped, hey, don't worry, you can do oncology later. After postdoc, you can go there and you can, I mean, he kind of motivated. He's a very nice guy. So then that looked me very challenging. I took that project. So we started analyzing all the sequences of Staphylococcus aureus and why there is no vaccine candidate so far, because it is known from it's a dangerous organism and it has a multi-drug resistance. Even vancomycin also, it only vancomycin for the multi-drug resistance stuff, only vancomycin will act. So there is a huge unmet need for these patients, especially end stage renal dialysis patients or old people who have fractures. I mean, they put steel rods in their bones. I mean, they get infected, then they have to be removed. So, and it's surgical wounds also if they get infected with this. High unmet need is there. So we were that time, even we thought of doing chikungunya also, because there is no vaccine for that, dengue and chikungunya. But due to some these infectious disease controls and regions, we didn't want, Dr. Khatri didn't want to do on these viruses. So, and also that time in 2000, this is the period of 2001 and 2. So you remember 9/11 and their white powder
Anthrax.
thesis and going on.
Mm-hmm.
So we wanted to do on Bacillus anthracis also.
Mm-hmm.
So I suggested that. So he agreed. So he said, and Dr. Krishnella said, don't exactly do it on both Bacillus anthracis. Take 2 or 3 projects. We will see after 1 year, whichever goes first, we can do that. So, so I proposed a thing called cell lytic enzymes. So it's present, I mean, with my, I mean, I was not expert that time, but I was learning all these things, how immunity developed and how vaccine nature is there. So I realized to be a good vaccine, it need to be on cell wall, so that body starts producing antibodies, and if you induce that, so that's what I realized. I looked at all the vaccine candidates available, so I did analysis of the sequence analysis and all that. So we realized it is a surface protein would be a good candidate for vaccine. So then I also, when we were looking at lysostrapin is also it clears the cell wall. I mean, it lyses the cell wall. That's how Staphylococcus was killed. So I mean, I was thinking it's kind of a double-edged. I mean, if you look at some autolysin or lysine enzyme, if you put it on the bacteria, it will die. At the same time, since it is a cell wall protein, our immune system might recognize it as a potential antigen and create an antibody. So they liked that idea, both Dr. Khatri and Dr. Ella. So for Bacillus anthracis, I identified a novel protein called amidase. It clears the cell wall. So we cloned everything, and I mean, Dr. Khatri and Dr. Ella, they had some collaboration with NICD, in Delhi National Institute of Chemical Disease, communicable disease.
Yeah.
So they provided Bacillus anthracis DNA, genomic DNA. They said we can't give anthracis. Then we asked, why don't give us anthrax as a bacteria, but at least give us a DNA. I mean, for us, only genomic DNA is enough. We don't need a bacteria itself. I mean, we proposed all this. I mean, we are doing, and it's a nice proposal. So they agreed, and they gave genomic DNA. So from genomic DNA, I cloned a protein from, gene. It's called amidase. So we did all that, and then we sent that purified protein to them. I mean, we need to know it's a functional also, right? So they only tested and it is working. They said, when you put this protein on bacteria in high quantity, so bacteria will die. So they said, oh, it's working. So we thought of everyone was happy, and we want to see that as a vaccine candidate. So that time there is a publication in Science on the same protein.
Okay. The
So,
scoop.
I was so disappointed.
Mhm.
So it's just by till that time I was 8 months to 1 year into my PhD.
Mhm.
So I was so disappointed. Then Dr. Khatri and Dr. Rela said, "Come on, man, you have this much brains.
Yes, absolutely. Yes.
Why are you disappointed? Don't worry, you will do it something else, or maybe we can characterize differently." Then I lost interest. No, they already published, means they would have done a lot of work by this time.
But
So
here, one thing which I would like to ask for on behalf of the audience which are watching this, that what we have heard till now is like the struggle, like without appointment reaching to meet Dr. Brahmachari, that struggle and the environment which was not suitable. You didn't like it, you moved it out. Taking those challenges, okay, the struggle and the challenge and the
mm,
achievement which we can see right now. So your journey is from a medical shop to thinking to develop into a diagnostic lab, and now you are in leading, CRO company and doing oncology, exactly what you wanted. When you summarize this, and then uh, now when budding researchers are coming up, okay,
mhm.
they always see that, okay, things don't work this way, and there is always a possibility to change the field as you have done.
Yes.
What are your thoughts or suggestions for these researchers who are very enthusiastic, not 100% sure that what they are interested in, and
Uh-huh.
how they should approach it?
Wow, that is in my view, actually nobody can pinpoint that, but
Mm-hmm.
I can only share based on my experience.
Mm-hmm.
So, see, whatever I'm doing currently, I'm enjoying thoroughly. In last 5 years, in my clinic, my expertise in my work in clinical research, I can say 3 or 4 drugs are being FDA approved. I didn't do any work on that drug as such directly. I was not involved in the development of it. I did not make the drug, or I did not make the vaccine or medicine, but I was involved in the process of reaching it to the people. It was
Mm-hmm.
discussed in the laboratory, and some pharma company bought the license, and this pharma company want to bring it to the people, and also, of course, they will make tons of money. That is a different thing. So, current FDA or US guidelines is that once it is discovered in lab, it has to come to the market. For the people use, it has to go through the process called clinical trials, and it has to be approved. They FDA review the clinical trial data. If they think the benefits are higher than the risk, then they would give approval, and then company would able to sell the drug to the in the market. So I was. I'm only one employee out of maybe 1,000 employees worked in this
Mm-hmm.
process. So, but I still feel very gratified. My name will not be there on anywhere. That company would acknowledge whole team as the CRO company, who this company helped them in more completing the clinical trial process.
Mm-hmm.
But I feel so fulfilled. I feel that my work is meaningful. I mean, two of the drugs, two oncology drugs were already approved, and they are working on patients. They are having their quality of life is improved. Oncology patients, their quality of life is terrible. They have always pains. They have to go through so many chemotherapies. They have to go through so many things. And in a year, they will go at least 100 to 150 days to the hospital. They don't stay in hospital, but they have to go so many times to the hospital, and their life is extended, so their quality of life is improved. So I feel that something I have my work. I was involved in this process. So in that way, I feel, of course, the rewards are also great. As a compared to my academic settings. So See, this is a mixture of once you realize academics are enough based on your lifestyle and family needs, and for me, uh, I wanted to become my original choice as a faculty, and I want to become a professor at university and have research grants and establish my research projects and develop a few molecules and have a patent and have a start my own company or give license to that. That is my dream, original dream, but I could not get it. But that there are so many
You
reasons.
can still do that. And that's what we have seen in
Yes,
researchers,
of course, I
that
would.
out of their work they are becoming entrepreneurs. And this is one thing which is very fortunate for all the young researchers who really want to make their thoughts so clear from the start that what they want to, and then they just work towards it. And we have stories that how even PhD students, out of their PhD, they started their companies. So
That
there are so many stories.
I'm glad to hear those stories, but I could not do, unfortunately. But I would do certainly at some point of my life.
Absolutely.
Yeah.
Absolutely. So here, one more thing which we would like to say, that contentment, and this is for all the audience also, that the contentment it gives, either you are doing a translational research as Dr. Narayan is doing, and you can see how content he is, that how happy he is that he was involved in the process where patients are getting the benefit of his work. Even if you're involved in fundamental research or the foundational research, which Dr. Narayan has done for a long time in labs, this is what is more important, because every researcher want to help human, in public health or in any other way, as Dr. Narayan does for their improving their lifestyle. Even sometimes just getting rid of pain is more than enough for them because they are suffering so bad. So, Dr. Narayan, we are walking towards the end of the conversation. I would like to ask the major role of family and friends in your research career.
Uh, of course, my family is very, very supportive. My friends are very, very supportive. But I mean, the thing is, the decisions what I made sometimes they didn't like. So even though they didn't like, they supported me because I mean, I love my family a lot. My family loves me. So in either my parents, my family, or way back in during PhD, before PhD, and none of my family members are science people. I mean, they're all business people or different kind of category. They don't even understand DNA. I mean, they know what is DNA, but they don't understand a test tube or science experiments. But they supported me because I had passion. I mean, I love science, so they said, "Okay, do whatever you want." And later, my wife also, she herself is a scientist, so she also supported me all these changes and all this process. And in fact, even she also moved to the similar field because of variety of reasons. So I wanted to add something when you said about translational, foundational research, and all that. So see, when I was doing my basic research or science foundational research and all that, emotionally I was very much satisfied. I discovered so many things. I mean, I made a vaccine candidate, I got a patent, and for some reason it was not developed due to scaling up issues. And later I had discovered a pathway in the neurology, in the Alzheimer disease and the neurotoxicity. And then later, because of my visa issues, I had to take whatever role I got in US. So I had taken infectious disease in virology and all that, respiratory infectious diseases. I had discovered at least three pathways which are very, very important in oxidative stress in lungs, in respiratory viral infectious diseases. I had nice bunch of high impact publications. So emotionally, the emotional passion of my science, it was highly satisfied there. I wanted to become faculty, but I could not. But now, where my financial need were not satisfied by doing always doing long time in foundational research because of the position. I mean, I could not become faculty. I was worked always as a research scientist in a university. So then I moved to translational or whatever is going on. It's a clinical trial research. So it's moved about 5 years ago. Here, my both financial and emotional needs also satisfied. I mean, because of my contribution, about at least 2 drugs were approved, and also rewards are also high. So it all depends on each and every person's their needs. I mean, both are good. I mean, you can't say which is good and which is not good. It is everyone's individual choice whether they want to be always in the foundational research, or I mean they want to move to an industry and pursue career. And in industry also they do great. Without industries we would not have medicine. So
Absolutely. Yeah,
so
well
we don't
said. Well
have a
said.
negative impression on industry.
No, absolutely. It was pleasure having you, Dr. Narayan, at the platform. And at the end, we thank you for your time and sharing this journey, which will ignite so many, researchers now to think and to follow their passion to the T, that nothing can stop them. And you are live example of that. With this, we would like to say well wishes for you for, you can start a company and can do what you wanted to be. Thank you very much.
Thank you. Thanks for having me, Raja.